I'm Eric Topol, and I'm really pleased to have you joining us for what I think is going to be the culmination of a great couple of days with this Spectrum Symposium. It's a session that I've been really excited about for the months since we planned it, and it's really putting together the COVID pandemic story in just a couple of hours with some extraordinary people. Two of them I've gotten to know through the pandemic as Professor Akiko Iwasaki at Yale, who is an HHMI professor there in immunobiology in the medical school in the School of Public Health, and Professor Angela Rasmussen. Angie is associated with Georgetown on the faculty, but soon to be moving to Saskatchewan to the university there, the Vito, one of the top research centers in the world. And my friend Peter Lee, who is the head of research at Microsoft. And we're going to be serially going through those three topics of immunology, virology, and then AI and big data. We'll then have a panel, and Christian Anderson, my colleague at Scripps Research, will join us, and we'll take some of your questions. I've got so many questions for these folks. It's going to be hard to get a lot of yours in, but it's going to be a fun session. And this genes, vaccines, machines is kind of bringing it all together. How much we've learned already a year into the pandemic, and how much more we still have to learn to get through it. So let me turn over to Akiko. Welcome, and we'll look forward to getting your comments about the immunologic aspects of COVID-19.
Thank you so much, Eric, for having me on this panel. Really delighted to be here with Angie and Peter. So today I'm going to focus on the immune response to SARS-CoV-2. One thing that we're learning about the COVID-19 disease is that it has a very large distinct outcome of infection depending on the host. And in some people, it causes asymptomatic infections or mild infections. And while others suffer from moderate to severe and sometimes even lethal diseases. And others also have acute disease that they can clear and recover from, while others suffer from long haul disease that are suffering from months of symptoms. And so currently, we don't understand what dictates the outcome of COVID-19 disease in different individuals, but it certainly has things to do with the host, such as the age, sex, comorbidities, as well as pregnancy status and other issues associated that might impact the immune system. So today I'm going to give you a couple of stories from my own group that demonstrate the distinct immune responses that occur in moderate and severe diseases, as well as touch on another story that has to do with potentially long-term consequences that can occur from COVID-19 infection, which has to do with autoimmune disease. So the first story that I'd like to share with you was led by these four amazing trainees, Carolina Lucas, Patrick Wang, John Klein, and Tiago Castro. And this has to do with longitudinal immune profiling of patients that came in through the Yale New Haven Hospital and their virological and immunological analyses. So the way in which we did this study was to recruit patients who had either moderate disease or severe disease. And these severe and moderate diseases were further divided into 6 different scores that we assigned depending on the amount of supplemental oxygen that people required during the hospital stay, from no oxygen to 6 liters per minute to mechanical ventilation and to death in the case of score 6. And we also recruited uninfected healthcare workers at the same time to be able to compare their immune responses in immune cell types. So by using multi-parametric flow cytometry analyses, what we noticed was that in COVID patients, especially in the severe patients, we saw a decline in the T cell, both percentage as well as number, and also increase in inflammatory monocytes, as well as increase in these appearance of these low density granulocytes, which are normally not found in the PBMC fraction, but are coming into these fraction in the case of COVID patients. And these included neutrophils and eosinophils. And this really correlated with the disease severity. And what appears to distinguish the severe patient from the moderate patients are the viral load in the nasopharynx that were monitored over time. So here in pink, I'm showing you severe patients' viral load from the nasopharyngeal swab over time in the hospital. And what you see is a maintenance of high load of virus, whereas the moderate patients were able to clear the virus eventually to below the threshold of positivity. So this indicated that there is this distinct feature associated with the severe disease, maintaining this very high viral load. And we also saw that this viral load appears to be what's driving the interferon and cytokines, and not the other way around. These antiviral cytokines, such as type 1 interferon, interferon alpha, or type 2 interferon, interferon gamma, are not able to reduce the viral load, but are driven by the viral load, which is shown here on the x-axis. We're plotting the viral load. And on the y-axis, the cytokine levels, and you see that there is a positive correlation between the viral load and the cytokines. That extended to things like this inflammatory cytokine TNF and TRAIL. And we took this one step further and asked, what are the level of these cytokines within the first 12 days since symptom onset in patients who died of this infection, which are indicated in pink, and those who were discharged from the hospital live. So these are the navy circles here. And what you see is that there is an elevation of interferon alpha 2, as well as IL-1 receptor antagonist, which is indicative of IL-1 receptor signaling in the patients that have ultimately died of this infection. And those cytokines were elevated within the first 12 days of symptom onset. And we saw a similar trend of elevated cytokines for other cytokines as well. And so this is a slide that I borrow from my lectures that I usually use for teaching immunology, because in our class, we teach that distinct types of immune responses are induced depending on the type of pathogens that we encounter. For instance, for parasite response, we mount Th2 immunity. For viral and intracellular bacteria, we mount Th1 immunity. For antifungal immunity, we need Th17 cells and regulatory T cells in order to suppress a hyperreactive immune response. And TFH is induced for B cell help. And this is dictated by the interaction between the dendritic cells that presents the antigen to naive T cell. And the fate of the T cells are dictated by the first order cytokines that are secreted by the dendritic cells that program these naive cell into these distinct types of effector lineage. However, what we found with the severe COVID is that all of these arms of the immune system is engaged. So all of type 1, type 2, and type 3 immune responses are elevated as they progress through the disease. And again, this pink indicates the severe patients and their levels of monocyte IL-12 interferon gamma for the type 1 immunity. For type 2 immunity, we're plotting things like eosinophils, eotaxin, IL-4, IL-5. These are the typical type 2 immune responses that are becoming elevated over time in the severe cases, whereas in the moderate patients, they either stay low or just declining in some cases. And we also saw even elevated IgE levels, which is quite unusual for an antiviral immune response. And again, we saw all kinds of type 3 immune effector functions being engaged in severe COVID as well. So we took the plasma cytokines and growth factors that are measured from the patients, both moderate and severe, and we asked whether they can be clustered based on these levels of the proteins, cytokines and growth factors that are found in the plasma. And essentially, there was 3 clusters of patients. So each of these columns are belong to a patient, and each row represents different cytokines and growth factors that are listed here. And what we saw was that the cluster 1 patients had an enrichment. So the darker the color, the more higher the levels of these cytokines found in their plasma. Cluster 1 patient really had an enrichment in tissue repair growth factors, EGF, VEGF, IL-7, and so on, that are elevated in their plasma. The cluster 2 patients, in addition, had these elevated chemokines, as well as mixed cytokines that belong to all 3 effector arms. And then cluster 3 patients had a very dark signals here belonging to type 2 and 3 effector function, as well as mixed cytokines. And so what does this mean? When you follow the trajectory of patients that belong in clusters 1, 2, and 3, what we see is an interesting divide in that the cluster 1 patients, they had some clinical scores to begin with, but they were able to reduce their clinical score and are discharged out of the hospital. Whereas cluster 2 and 3 patients, they had time-dependent elevation in the clinical scores, and they had elevated frequencies for coagulopathy and mortality compared to the cluster 1 patients, indicating that these 3 clusters that were defined based on the plasma level of cytokines really were revealing something biologically relevant, in that the first cluster of patients who had these tissue repair growth factors went on to recover from this infection, while the other 2 clusters had worse disease outcome. And when we took all the parameters that we measured for all the patients and asked for the biomarkers for COVID-19 mortality, what we detected is that the top biomarker, it turns out to be IL-18, which is a cytokine released as a result of inflammasome activation. And the second top mortality biomarker was interferon alpha itself. And then other factors are listed here. So what these data showed us is that there is a persistent viral load that's detected from the severe disease patient. And this viral load is what appears to drive the interferon and cytokine responses. An increase in these levels of cytokines and interferons seen within the first 12 days of symptom onset were elevated in the people who died of this infection. And there are 4 signatures of protein that distinguish patients who recover versus who go on to develop worse disease outcome. And the worst outcome was found in the patients who developed these mixed cytokine responses, and which we would consider them a maladaptive response to a viral infection. So putting these data in the context of what we understand about viral immunity in general, we have a hypothesis that, the earlier you can generate the interferon, the better outcome it is for recovery from that infection. So here the viral load comes up and this person is able to generate interferon response and clear the virus, recover from a mild disease. And this might be happening in young individual as well as low viral exposure settings. On the other hand, patients that we were studying, which is really this late time point, when we had this elevated interferon response with elevated viral load, this really is indicating that delayed interferon response is actually more harmful to the patient, and that this is resulting in the severe disease that we see in older adults in high viral exposure settings. We also know from work by John Laurent Casanova's group that there are patients who have deficiency in the genetic mutations in the genes that are involved in interferon signaling as well as induction. And those patients have very little interferon. And in parallel, there are patients who have neutralizing antibodies to type 1 interferons who also have functionally very reduced interferon in their system. And in these settings, you have uncontrolled viral load and severe to lethal COVID that happen. And finally, I don't think we're powerless against this virus. We can maybe interfere with all of these processes by injecting interferon early during this infection. And this has to be, it's a little tricky because for this virus, the symptom only begins after the peak viral titer. So how do we catch this? We might have to think about prophylactic mechanisms for this. But essentially, early interferon should lead to a better outcome. So in the last part, I wanted to just touch on our recent published work on autoantibodies in COVID. And this was really led by an amazing colleague, Aaron Ring, who's in the same department as I am, with his trainees as well as my trainees. Together led this work. And the impetus for this looking for autoantibodies was that we know that in some patients, COVID-19 has very long-lasting symptoms, as I mentioned earlier. That involves so many different organ systems. And so we thought that perhaps autoimmunity generated against different tissue antigens might be driving these types of multi-organ symptoms. So Dr. Ring's laboratory developed this new technology called Rapid Extracellular Antigen Profiling, or REAP, whereby he has generated a yeast display library encoding over 3,000 extracellular or secreted proteins from human that's displayed on the surface. And so we can simply take our patient's serum and then add it to these yeast library and pull out the ones that are being bound by the antibodies and to identify the autoantigens that are being recognized by patients. So using this REAP technology, we were able to identify multiple diverse array of autoantigens that are found in COVID patients. And here again, each column is a patient. And I've divided it into severe, moderate, mild to asymptomatic patients. And the brighter the yellow, the more autoantibody there is in a patient. And each row represents an antigen. And the first thing you notice here is that this panel here, representing type 1 interferon and type 3 interferon, there's a lot of autoreactivity in the severe COVID patients, indicating that this is really consistent with what Jean Laurent's group also showed, that in the severe COVID patients, there is an enrichment for people with autoantibody to type 1 interferon. And you can see that there are antibodies against all kinds of immune cells, as well as other inflammatory factors that are found. And so all these yellow boxes that you see are representing autoantibodies in a patient. And these autoantibodies to interferon likely has a functional consequence, because when we follow over time, the patients with these anti-interferon autoantibodies are unable to clear the viral load, whereas those patients that are matched with severity, sex, and age are able to reduce the viral load over time, who didn't have these autoantibody to interferon type 1. In addition, there are many examples where we found that autoantibodies to cell surface markers for a variety of lymphocytes exists. And this is just showing some evidence for patients who had autoantibody against B cell surface markers. They had very little B cells in circulation, and they also had no ability to mount an antibody against the SARS-CoV-2 RBD, indicating that functionally, these patients are defective against in B cell response. In addition, we found a whole range of autoantibody against wide tissue antigen targets. So here, again, I won't have time to go through each of these antigens, but just to say that within the CNS, vascular, connective tissues, saliva, skin, GI tract, and so on, there are these antigens that are being recognized by antibodies in the serum from these COVID patients. And they may have functional consequences. We are currently trying to validate some of these hits. So this part of the work demonstrated that there's diverse autoantibodies found in COVID patients, and some of these autoantibodies appear to be pre-existing, especially the ones against the interferons. And these interferon type 1 autoantibody appear to impair viral clearance in the patients. And there are other autoantibodies against immune cells that appear to diminish the number of these cells as well as their function. And there are these autoantibodies to tissue antigens that may contribute to significant tissue-specific dysfunction. So I wanted to kind of end with this hypothesis where we are seeing some long COVID patients feeling better after vaccination. These are still, the numbers are low, and these are mostly patient-based groups reporting 30 to 40% of the long haulers feeling better after vaccination. These are not controlled trials or anything, but at least reporting suggests that there's some benefit to vaccination. And so I believe that the long COVID can be caused by either persistent viral reservoir or autoantibody response or autoimmune T cell response. And vaccines may improve the long COVID symptom by either eliciting a robust antibody or T cell response that can target the viral reservoir, or the innate stimulation by these mRNA vaccine, for example. The mRNA itself is a ligand for various viral sensors in the cell. And that can trigger cytokines and interferons that may divert the autoreactive T cells and B cells away from this, the toxic effector functions that they're conducting. So trial and analyses of long haulers before and after the vaccination will really provide the much needed insights into the disease as well as potential therapeutics for the long COVID patients. So I will end here by thanking my amazing lab members who are really working around the clock trying to understand immune response to COVID, as well as implementing testing in the community. And none of this work could have been done without the Impact Yale team, which was established by the investigators led by Dr. Albert Ko and many others listed here. And here are many other authors, which I didn't have time to acknowledge, who contributed to the work that I was able to describe today. So thank you very much for your attention.
It's always a tough act to follow Dr. Iwasaki, but I'm always glad to do so because I think that our work is actually really, really complementary. And I'm pleased to see that since the last time I heard her speak, I see that her hypothesis about the role of interferons in the host response actually hasn't changed that much, and it still complements mine. So that's what I'm going to talk to you about today. Since I study the host response to SARS coronavirus 2, and even though I am a virologist, I tend to focus on the role of the host in viral infection and how that host response contributes to disease. But unlike Dr. Iwasaki, I'm not just focused on the immune system. I focus on global host responses. I'm also going to talk a little bit about how we can use those for diagnostics, prognostics, and potentially drug discovery as well. So before I get started, though, I'd like to begin with my territorial acknowledgement and equity statement. And today I'm in Seattle presenting from the unceded ancestral homelands of the Duwamish people. The work that I'm presenting today was performed on the lands of the Duwamish and Puyallup people here in Washington State, as well as the Lenape people of Manhattan Island. I acknowledge and honor the first people of all these territories and their tribal governments, as well as their history in caring for these lands. And I'd also like to acknowledge the long history of systemic inequity in academic science that really spans centuries. My prior institution, where much of this work was done, Columbia University, and my current institution, Georgetown University, were both founded using profits from the transatlantic slave trade and the sale of enslaved people. And in addition, those institutions, as well as many others, excluded women and people of color from the academic community for more than 200 years. And this has really left a long and painful legacy of racial and gender-based inequality that continues to this day. So I encourage all of you to consider how you can contribute to making scientific research a more equitable enterprise going forward. And with that, I'm going to begin by telling you about the approach that I take when looking at these global host responses. So I use what's called a systems biology approach, but that doesn't really mean anything, because if you ask any systems biologist what systems biology actually is, you're probably going to get a different answer from each person. So this is kind of what this means to me. This is really an iterative, cyclic approach to developing hypotheses, testing them, generating new hypotheses, and then testing those. And we start with what most of us would think of when we think of lab work, virology. So this is experimental models, whether that be cells in culture or animal models. You infect them with virus and you conduct normal virological, biochemical, immunological studies on them. Then I use these omics-based approaches to kind of get a global lay of the land in terms of how the host is responding to that infection. Most often I use transcriptomic approaches or total RNA-seq to look at the entire gene expression landscape induced by the viral infection. We use bioinformatic and computational modeling approaches to make sense of all of that data. And then we can start to use that to develop hypotheses, and also for some translational applications, such as developing biomarker panels, identifying novel drug targets, or looking for correlates of protection for vaccines. And really, this combination of multiple experimental systems can address some really complex problems, such as those involving the host response, because the host response has a lot of different moving parts. And really, when you think of pathogenesis and how viruses cause disease, there's really two sides to this story. On one hand, you have the virus, and even more complex RNA viruses like coronaviruses are still relatively small compared to the host in terms of their genome size, the number of genes that are encoded, and the function of those different proteins. Even though they're all very multifunctional, there are far fewer of them than in the host. And in this case, if the host is us, that means we have a 3.2 billion base pair genome that's organized into roughly 20,000 genes, compared to a coronavirus's 26 genes. These genes are all organized into multiple specialized pathways. They're expressed as multifunctional proteins in multiple isoforms. So the host side of things is very complex. Not to say that the virus side isn't complex. Of course, it is too, complex in a different way in some senses because of the simplicity of viruses relative to the host. But there's a lot of moving parts on both sides of pathogenesis. And that's what I'm going to focus on. And we've known for a long time that host responses can determine pathogenicity. This is just two examples out of many. Influenza viruses, different influenza viruses induce very different gene expression profiles. In Cynomolgus macaques, in this case, infected with a seasonal flu versus a high pathogenicity avian H5N1 flu. We see the same thing in Ebola virus, which is highly pathogenic in people, versus Reston virus, which is not in macrophages. So we can find these differences in the host gene expression programs that viral infection induces, and we can link this to pathogenicity. So how can we use this information to diagnose infection, predict outcome, or identify new therapeutic targets? Now, talking about SARS coronavirus 2, very early on in the pandemic, so this came out, I think, last May, Ben Tenover's group at Mount Sinai did a really interesting study in which they looked at host responses in cell lines, in ferrets, and in COVID patients. And what they found was that in the lungs, SARS coronavirus 2 infection profoundly suppressed type 1 and type 3 interferon responses. They hypothesized that this led to the cytokine storm that Dr. Iwasaki talked about a little bit. And we just saw how the timing and magnitude of the interferon response is really important in determining ultimately the clinical outcome. We also saw from this work in Ben's lab that SARS coronavirus 2 really does induce unique host response profiles compared to influenza A, for example. This is both in terms of the overall transcriptomic landscape, as well as the types of pathways that are being induced. So it really is fundamentally different from other viral infections, including other viral pathogens that infect the respiratory tract. So I decided to take a look at this in the rhesus macaque model, and this was developed by my collaborators at NIH Rocky Mountain Labs. This was Emmy DeWitt and Vincent Munster's work, in which they infected rhesus macaques with SARS coronavirus 2. These animals develop observable lung lesions that can be seen at necropsy as well as by histopathology. You can also identify areas of infiltration by X-rays. And in this case, though, we looked at the whole blood. So the disease is occurring in the respiratory tract in the lungs. This is a model of moderate COVID-19. So these animals don't actually get that sick, but they also don't have any virus in their blood. So we looked at blood samples. So this is really looking at systemic responses rather than the responses of the cells that are directly infected. And what we found, and I'm going to go through this very quickly, is basically this is pretty typical of non-human primate experiments. When you're looking at the overall transcriptome, there's a lot of noise, basically, with what we're seeing. And we saw most of the gene expression between days 1, 3, and 5. Post-infection, this peak of gene expression at day 12 is probably driven by 1 or 2 animals. This is because these macaques are quite genetically distinct from each other, so we normally expect to see a lot of inter-animal variation. But we can still find some pretty good information from looking at the early time points in these animals in terms of what's being upregulated, downregulated, and how these are all shaking out in terms of the biology that's going on. So what we saw in the blood, and again, these animals have no virus in their blood. So this is looking at the systemic responses of cell or not upregulation in interferon, inflammatory genes, and interferon stimulated genes. This really suggests that systemically there's a rapid response in this model. And again, this is consistent with what Dr. Iwasaki was talking about in that people who have a rapid interferon response typically have more desirable disease outcomes. They have less severe disease. That's really consistent with what we're seeing in these macaques. The serum cytokines from these animals were consistent with our findings. And also we see similar things in terms of the immune cells that are driving these responses. Early on, we see increased myeloid and phagocytic cells, but we don't see sustained levels of this. So this is consistent with these types of cells that are being mobilized to go to the site of infection and clear it. We also, over time, see an increase in plasmacytoid dendritic cells that are very specialized to secrete large amounts of type 1 interferons. So we see, again, in this moderate model of COVID-19 disease, that we are seeing rapid and robust interferon responses along with controlled inflammatory responses, and that might be linked to this moderate disease outcome. So this is consistent also with what's been observed in human patients. This is another study from last year in which they looked at patients with different levels of disease severity, and they specifically looked at interferon and ISGs in the peripheral blood of these patients. And they found that patients who had severe or critical COVID-19 had far fewer interferon and interferon stimulated genes being expressed. And this was pretty significant compared to mild and moderate. So my hypothesis, again, agrees with Dr. Iwasaki's in that these systemic interferon responses, their timing and their magnitude probably determine disease severity. So while in the lungs, the cells that are directly infected, you will see interferon suppression, and this is a normal feature of most viral infection, you will then see severe or increased inflammation in those tissues. Systemically, in mild and moderate disease, you will see these early rapid interferon responses and controlled inflammation that lead to viral clearance in the infected tissue. And in severe cases, you would see suppressed interferon responses or delayed interferon responses along with uncontrolled inflammation. Now, we do need to test this hypothesis in animal models of differential COVID-19 severity. So that's something that I plan to do once I get settled in at my new institution. Now I'm going to talk briefly about how we can take some of this data and combine it with data that's already in the public domain, or data that in this case I generated with my collaborators prior to the pandemic, and make some more use out of it. So prior to the pandemic occurring, I was working on a project with Vincent Munster and Dick Bowen and Ben Garcia looking at tolerance, what we call tolerance in MERS coronavirus infection. And this is basically why some species or some individuals get MERS and have very mild disease, whereas some others end up developing very severe disease. And we looked at this across multiple species. We have a dose-dependent model of tolerance in mice. We also looked at marmosets, which develop severe and lethal disease, as well as dromedary camels and alpacas, which are tolerant to MERS infection. And then we did this same type of transcriptomic work, and then we decided that we wanted to use machine learning approaches to try to predict outcome. So the way that this works is you take host gene expression profiles from these animal models, you go through these gene expression profiles and you iteratively pick these features, in this case, genes that are upregulated or downregulated, and you see how they perform in a model as far as being able to classify a particular outcome. So we wanted to know if we could take host response data from animal models and use it to predict outcome in human patients. And when we did this with the mouse model of MERS that I just described, we found that we were able to predict outcome about 90% of the time correctly. But again, this is in mice. How would this work in people? Well, we already did this also with Ebola virus. So we have another model of differential Ebola virus disease that's a panel of genetically diverse mice called the collaborative cross. In that model, we were able to predict Ebola outcome with almost 100% performance using the mouse data alone. Then we took a data set that was available publicly that looked at West African Ebola patients, 24 of whom survived Ebola virus disease and 88 of whom were fatal cases. This had already been used by the people who published this to use a similar approach to predict outcome, and they did so with about 79 to 85% accuracy, depending on what algorithm they were using. So we used this to test the model we had trained on our mouse data, and we found that it predicted outcome correctly about 75% of the time, which is good in terms of proof of principle, probably not good enough for the clinic. But once we added data from non-human primate data set that came out of work that we did with Tom Geisbert at the University of Texas Medical Branch, Galveston National Labs, we were able to boost that performance up to about 96%. So we were able to correctly predict outcome 96% of the time if we were using data from mice, from monkeys, and from people. So we thought this was a pretty good indication that we might be able to take data from other coronavirus models and supplement the limited amount of data that we have for SARS coronavirus 2 and MERS coronavirus with other bits of data. I've also been working with Michael Kirby at Colorado State University, and he's a mathematician and has made these sort of bespoke machine learning models that can classify people and whether or not they're going to shed virus and infections, right down to the hour at which they begin shedding with several different viruses. So we know that this can be done. We've been able to do it now with our animals and with humans. Michael has been able to do it in his group using these custom approaches. So we plan to do this in terms of coronavirus pathogenicity, and this hasn't been done yet, but it's in store. So the idea here is that we would take transcriptomic data from the MERS coronavirus animal models that I was talking to you about, from the SARS coronavirus 2 animal models that I just told you about, as well as some others that are in the public domain, and then mix these up with data that's there already from other highly pathogenic beta coronavirus infections. So SARS classic, MERS, SARS-CoV-2 that are in databases like GEO. And by developing these pan-coronavirus classification signatures, we can not only maybe predict pathogenicity haven't even been discovered yet, we might also be able to predict, the wildlife, whether they're even competent human pathogens at all. So we think this really does have pretty good applications in terms of pandemic preparedness, as well as developing prognostics for future coronavirus pandemics or other virus pandemics. And the nice thing about working on the host response is that in this case, when you're dealing with a human host, the host never changes. It's just the virus that changes. So if we can find some common threads between viruses that have similar features or are related to one another, we think that this will have applications long beyond the current pandemic. Okay, so as I said, my grand scheme of things is to integrate this data across these different platforms, experimental platforms, as well as these different pathogens, and kind of develop universal mechanisms of susceptibility and tolerance and predict pathogenicity or protection across species. And I think that I'm getting a little notice that I need to start wrapping up. So I'm just going to go very quickly through the upstream analysis and drug repurposing work that we've also done. So the idea behind upstream analysis and drug repurposing is that you take the transcriptomic data that you're seeing, but even though it's a great way to look at global responses, it's not necessarily a very good way to see what's not being regulated transcriptionally. So if something is upstream and is responsible for this downstream gene expression pattern, you might not see it in your transcriptomic data. So we use a different module of our functional analysis software that predicts what these upstream regulators are so that you can identify them even if they're not showing up in your transcriptomic data set. And we've done this again with our Ebola model, where we were looking for upstream regulators that are drugs that would either inhibit signatures associated with lethality or activate signatures associated with tolerance. And we identified 4 different drugs that met this criteria, including corticosteroids. Then we confirmed this with another database just to make sure that this wasn't an artifact of the particular software that we were using. And we found again that these corticosteroids were identified. So we tested this in the mouse model that we have. And while it wasn't necessarily something that you would recommend that people with Ebola go out and start taking dexamethasone, we did find that we could observe a significant delay in progression to death, as well as a delay in onset of clinical symptoms. So, oops, sorry about that. So we thought this was a good proof of principle. We've since done that with our MERS coronavirus data, and we were very pleased to see that dexamethasone again popped out of this. And as many of you know, dexamethasone is one of the few treatments that does seem to work for SARS coronavirus 2. So just finishing up here, we identified dexamethasone when we also looked at this with both the MERS data across these multiple species, as well as SARS coronavirus 2 data from ferrets, from human lung autopsies and biopsies, as well as the macaque data that I showed you earlier. So that was, again, really good proof of principle. Tocilizumab, which has also been shown to have a clinical benefit, also popped up here. So did beta estradiol, which may be important in terms of explaining some of the sex biases that have been observed in SARS coronavirus 2. So this is very crude early days of this. But again, I just wanted to show you because I think that there's going to be a lot of potential once we start integrating these data sets across different models of beta coronavirus infection. So that's just kind of what I've depicted here. And I hope that I've convinced you that you can use animal model systems and interrogation of host responses to look at many, many different aspects of pathogenesis. And there are many applications in terms of pandemic preparedness and response, including for wildlife, livestock, and agriculture, companion animals, and of course, people. And with that, I'd just like to acknowledge my many collaborators and look forward to discussing this all on the panel. Thank you so much.
So let me just start by saying, besides saying I'm Peter Lee, I was really impressed with both Akiko and Angie's presentations and learned a lot. So really appreciate the work they're doing. And I clearly need to follow up with both of them. What I wanted to do here is talk a little bit about the data analysis and particularly the use of machine learning and AI. And where that is going and maybe some both current and a little bit more future-oriented implications. If we just take a step back and think back over the past year, data has been pretty important in outbreak response. Of course, the whole world has become very attached to looking at the data modeling and forecasting work. And there have been many prominent organizations around the world that have been involved in that. If you look at the CDC leaderboard today, there are, I think, 56 or 57 such forecasting models, typically forecasting mortality and hospitalization rates, infection rates. Most of them are based in one form or another of SEIR, classical models, epidemiological models. But if you look at the typically the most accurate model, at least for a 2-week into the future forecast, it's a Microsoft model that actually is not based on SEIR, but is based on an ensemble of recurrent neural nets. And that tends even to beat the combination model that the CDC aggregates from all the others. And the use of recurrent neural nets, the use of machine learning shouldn't be too much of a surprise, but what's really interesting about that is it allows you then predictively to understand, well, if you were to impose specific restrictions, like a ban on bars and restaurants or a ban on large sporting events, it allows you to play forward the implications of that. And so machine learning has turned out to be important in that space. There has been, of course, a tremendous amount of work to bring AI and machine learning into both public health operations and into healthcare delivery. Patient number one in the US happened here in the Pacific Northwest, was treated by the Providence Health System. There were immediately a lot of predictive analytics and trend analysis machine learning problems that they brought forward since they saw clearly that there might be a crisis looming here. And even things like understanding the pre-positioning and the need for things like PPE ended up being machine learning problems. In the public health space, there has been quite a bit of work. The Microsoft HealthBot that was worked on with Providence and the CDC ended up being deployed at about 3,100 hospitals and health systems around the world and several governments. It provided very detailed machine learning in order to measure and do an agile response to ensure that, for example, that huge crowds weren't being brought into emergency departments or into nurse call centers. Even just slight word changes were determined in part by machine learning. And then finally, and I think the main subject of this session has been about research acceleration through the better ability to extract insight and knowledge from data. And there's been a tremendous amount here. And I want to point out that it's not just in understanding immune system response, it's not just understanding the molecular modeling and accelerating molecular dynamic simulations through machine learning, but even things like AI-powered chatbots that were deployed by companies like Walgreens ended up directing important numbers of people to key clinical trials. And so there was sort of an impact of AI at several different levels. But the thing I thought I would focus on first here is more on our understanding of immune system response. And I decided to focus on just knowing the other two panelists. It's also a little bit daunting because I'm a computer scientist by training, and Microsoft, of course, is a computer technology company. And yet what we found is that we were drawn into many of these research efforts around the world simply because of the need to apply more advanced machine learning to the problems. One of the projects we worked on that was started in November was with Adaptive Biotechnologies, which is a Seattle biotech company. And they had an idea, and we had an ongoing project that we had been working on with them for the past 2.5 years that was applying machine learning to the understanding of T-cell receptor repertoires. And in fact, in November, the ongoing diagnostic targets for that project were suspended. And instead, the total effort was directed to seeing whether there could be some sort of diagnostic support for COVID-19 using these techniques. So what is this about? Well, the overall goal of the project was to develop a platform for T-cell based multiplex diagnostics. And the specific goal in this project was to compute or impute a person's HLA types. Because HLAs present peptides to the T-cell receptors and the HLAs are highly polymorphic, we expected an individual's HLA types to shape in a profound way the T-cell receptor repertoire. And so, in fact, if we train our so-called type 2 models to predict specific HLA alleles using TCR repertoires, we do find a pretty small signal. Here's an example, in fact, then for A0201. So the procedure is pretty simple as a first cut. You take the first pass through the data and identify T-cell receptors that are in some sense statistically associated with the A0201 expression. And then you take a second pass and just simply count up how many A0201 associated T-cell receptors a person has. And so that's plotted along the y-axis, and then compare that to the overall diversity along the x-axis. And what you can see here on this chart is that this dead simple approach, it's just simple counting, there's no machine learning going on here, yields almost perfect separation. Now, this small training set of just, in this particular example that I'm showing, of 630 HLA type repertoires, we can get nearly perfect prediction for many of the alleles at all the classic class 1 and class 2 locations. And this is useful for a couple of reasons. First, any case control cohort that looks at T receptors should control for HLA types. And so with this approach, we can precisely construct appropriate control sets. And then second, this set of alleles includes many that are epidemiologically linked to autoimmune diseases. And so when we look at, for example, DQ2.5 haplotype, which is linked to celiac disease, or B27, which is linked to ankylosing spondylitis, and so on, we have a direct estimate of genetic risk for these diseases through this method. But perhaps most of all, what we're seeing is that we expect clear signal, and we are in fact seeing it. So that's comforting. Now, what I've shown here was training directly from the repertoires. And that's just, as I said, a dead simple thing. But in fact, this particular plot was just from one fold in a cross-validation run, and specifically trained on 503 repertoires. And now this chart is showing the results on 127 repertoires of the test set. It turns out, though, that we can get nearly comparable accuracy with a so-called N of 0 experiment, in which we train on 0 repertoires and test on all 630. Now, how is that possible? Well, we do this by, of course, using other data, specifically antigen-specific T-cell receptors. Our specific approach is, first of all, to use an assay at Adaptive Biotechnologies called MIRA to identify T-cell receptors that bind relevant antigens. Then we learn the antigen-specific models, and then finally we can scan the repertoires in the test set for TCRs that are predicted to bind to those antigens. And it turns out that if we choose the right epitopes, we can get a very strong signal. Now, in this case, we have only 3 epitopes chosen, the well-studied A0201 restricted immunodominant epitopes for CMV, for EBV, and influenza. And I'm guessing that almost everyone in this audience today has had an infection or been vaccinated against at least one of these viruses. And so if you express A0201, for example, there's a very good chance you will target the epitopes put into these models. And as you can see, we get pretty close to perfect positive predictive value. And the only real limitation is for some individuals who apparently either weren't affected by one of these viruses or mounted a non-standard response. Now, coming back to COVID-19, prior to the pandemic a year ago, starting with this concept, we started to look at other more complicated diagnostic targets, at celiac disease, at Lyme disease, at several cancers. And the reason that we felt that this was interesting is that we would be able to generate very large amounts of label training data for our machine learned models. We would not, for example, in the case of Lyme disease or a celiac disease model, need to find extremely large numbers of patients affected by those diseases. We would need just, we estimated, on the order of 2,000 to 3,000 affected patients. And then we'd be able to use this approach to boost dramatically the strength of the signal and the accuracy of the models using naive samples. And so that was the program that we've been on. And in fact, for several of those disease targets, quite a bit of progress was made. Then we hit the pandemic. And with the pandemic, a decision was made to suspend those efforts temporarily and focus entirely on COVID-19. And that commenced at the beginning of November, and last week we received the emergency use authorization for the T-Detect test based on this principle for COVID-19. And that test provides very high specificity and sensitivity for current or past COVID-19 disease or SARS-CoV-2 infection up to 120 days out. Now, taking a step back, I just wanted to comment a little bit about this sort of machine learning or AI-based approach. And let me just start by saying making a comment about the dependence on data. And this is something that came up in both of the previous talks. If we think abstractly, there's this thing called intelligence, and then there's a field of artificial intelligence, and it's not known today whether artificial intelligence, machine intelligence is a proper subset of the notion of intelligence or not, but there's some coverage there. Machine learning, of course, is a very small subset of the concept of artificial intelligence. And then within machine learning, there's yet a smaller subset, which is the idea of building self-improving machines that improve through the use of data. And then an even smaller portion of that is what we would call supervised machine learning. And supervised machine learning basically is data that has been labeled through human intelligence. And sometimes we talk about that being the digital exhaust of human intelligence. And for the most part, almost everything that is done in industrial research today, in commercial application, in AI, is based on supervised machine learning. What has been happening, though, is that there has been a revolution of practical application and scale in broadening from that red dot to that full orange dot, to being able to learn from data in an unsupervised way. And one of the examples of this has been happening in natural language processing. And so to explain how that's been working, let me show this test. This is a very simple fill in the blank test where you're supposed to guess the right word that belongs here. And this is in linguistics is referred to as a cloze test, C-O-L-Z-E. Cloze tests end up being very interesting because what you can do is you can form these cloze tests. So just gather all the text in any language from every digital source you can, the entire World Wide Web, any other place. Run it through a pre-trained multi-layer transformer to extract a contextualized representation that allows you to make a prediction or an attempt at filling in the blank. When you fill in the blank, of course, then you're able to assess how well you've done. And since we formed the tests, we created these blanks from all the gathered text, we're able to compute the loss based on that ground truth and feed that loss back into an improvement of the multi-layer transformer. And so with this process, what we're able to do is set in motion a process of training an extremely large machine learned neural model without any human intelligence supervision. We can just have a machine that, say, scoops up all of the text on the internet, just randomly constructs every possible cloze test, and then run this loop. Today there are, of course, quite a few academic researchers working on this, and there are two companies, Microsoft and Google, that have been working very hard to scale up the size of the neural model, sort of the parameter space involved here. And so here you can see what's been publicly disclosed. In fact, this is a little bit outdated because Google just publicly disclosed their 1.6 trillion parameter model. But we're rapidly approaching the multiple trillion parameter models. And this is, to give you a sense of the size of the neural nets, roughly 1 order of magnitude less than the number of neurons in an animal like an octopus. Now, there's been a lot of question about whether there's any hyperreality, and some of that's fed by some of the stunts that people show. One stunt is, you give 4 lines of Shakespeare to GPT-3, and then it just generates original text in the same style, or just out of the box, GPT-3 is able to just engage in reasonably intelligent Q&A with a human being. And furthermore, by doing a little bit of small task-specific training, you're able to get remarkably good performance out of these extremely large neural models for non-language tasks like image processing. This has been very important in the past year in being able to look at even clinical texts and be able to extract the meaning of clinical notes with remarkably little training, almost no training at all. But perhaps most interesting has been the application of extremely large neural models to fundamental questions in biology. The DeepMind folks made quite a bit of news with AlphaFold. But in fact, there has been here also an arms race, where the use of deep neural nets in order to do the static prediction of the static configuration of energy minimum configurations of proteins has been underway. And as the neural models have been expanding in size, the accuracy or the similarity between the predicted and native structure of proteins has been improving along with it. And again, this is really happening in part through a transfer learning effect from the natural language processing models. And while the model that DeepMind used for this is big, it's actually 2 orders of magnitude smaller than the actual hyperscale neural models that are currently underway in both Microsoft and Google. And so when you play that forward, and if you want to see, how far away we may be from being able to use machine learning, say, instead of molecular dynamic simulations in order to get a dynamic folding model, we're already within a few hundred GPU years of capability today. And with the next generation of models, we may be much closer than that. And so I think, if anything, what we've seen is that the use of machine learning AI has been very important today in the response to the pandemic, but the acceleration of application of this to problems in medicine and biology in the future look to me to be even more important. So with that, I will stop sharing and hand it back to our moderators.
Okay, thank you, Peter. I don't know any AI scientists who could also present immunology, so you got to get special recognition for that. Really great presentations, Akiko and Angie. I mean, this is a phenomenal substrate for us to discuss it. I want to bring along my colleague, Christian Anderson, for the panel, who's been doing, I think everyone knows, quite a bit of work in the genomic sequencing and trying to understand where we're headed with variants and so much more. So Christian, welcome. Best recruit I ever made in my career was getting Christian here from the Broad Institute, by the way. So let me start things off, and we'll take questions from our participants. But, both Akiko and Angie, you really delved into the interferon story quite a bit. And what's striking to me is that we've known about this. You've added a lot to the knowledge base, but we still don't have interferon as an early therapy, either prophylactically or, even at the earliest contact with someone with known COVID. And so it's really seems to me we've had so much work done on other drugs, but why haven't we made some headway in the interferon story? And if you could also comment, with those 10% or so people that have autoantibodies, would an inhalation of beta interferon, which is basically a repurposed drug, would it override that concern? So maybe Akiko, can you start with that?
Yeah, absolutely. So, as with all the other drugs, interferon has been tried in large randomized clinical trials. And as Angie really impressed upon us too, that there's a really important need to give the interferon early. Otherwise, we might be harming the people. And in fact, if you look at the recovery trial, which included all patients, including people who had severe disease with treatment with interferon, they did not see any benefit for that large cohort. And in fact, in some severe patients, it may have been actually a little bit more harmful than the placebo group. But on the other hand, there are other trials that included only mild patients or moderate early disease phase. And those trials are showing some benefit. So I think we really need to hone in on the right timing and the right stage of the disease. In particular, prophylactic inhaled interferon might be a really good thing to pursue in the future, especially for people who are in a high risk category, healthcare workers who are treating COVID patients and so on. And this can be even applied to other types of, respiratory viruses. And just to get to the interferon beta treatment of people who have anti-type 1 interferon autoantibodies. So we haven't seen anyone who actually developed antibody against interferon beta. All of these people are making interferon alpha isoforms, as well as some for interferon lambda, but fortunately, we have not seen any that made interferon beta. So I think that's a really nice kind of idea, Eric, that we might be able to treat people without knowing their autoantibody status, potentially with interferon beta early.
Yeah, be sure. Nice to get that into high gear, fast track it. Angie, anything to add on that?
I don't really have anything to add. I mean, I think that one thing that has always come up in every data set I've ever looked at is that even in the cases of severe disease, if you look at a later time point, you always see tons and tons of interferon. And I think a lot of people find that surprising because it's always thought that interferon antagonism is something that leads to pathogenesis, but it's not so much that there's no interferon at all. It's just that the timing of the interferon's not right. So I completely agree with Akiko that probably early treatment is the way to do it. And that would explain why you don't see an effect when you're looking at patients who are already very severely ill. I think that prophylactic interferon is an interesting idea, especially if it's inhaled, because the only other thing to consider about that is that interferon is expensive, and it also is, when given IV anyways, it's not well tolerated at all. And I did my PhD or my postdoc working on hepatitis C virus, and I'm old enough that when I was doing that, the primary treatment for HCV was pegylated interferon and ribavirin. And for genotype 1 infections anyways, it was so ineffective that most clinicians were not even prescribing it to their patients because it caused such serious reductions in quality of life unless they showed signs of progressive liver disease. So I think that if you can get interferon in a form that would be tolerable for uses of prophylactic, I think that's a really interesting approach to take. Otherwise, I think though that early treatment is going to be the way to go with interferon.
Right. Now, each of you have so many multidimensional contributions you made during the pandemic, which we could get into, and we won't have time, but I do want to get to Peter because last time we spoke, he and I were comparing notes about convalescent plasma. Yes. And for those of you who don't know, Microsoft was like the backbone of the national convalescent plasma. Over 500,000 Americans received convalescent plasma. So maybe can you just give a little bit of perspective about that, Peter, and kind of your latest sense of where that is going or has been?
Yeah, it's been quite an odyssey, and I sometimes think 10 years from now, I'll look back on this with some combination of post-traumatic stress and and pride. It is kind of amazing, and really credit to the people at Mayo Clinic and Meyer, Johns Hopkins, and so on, because they sort of spearheaded an early access protocol that enrolled the first 100,000 patients or so. I think that the medical science is difficult here. It's a subtle thing. There definitely appears to be very little benefit in late disease progression. So again, it's something that if it's to be effective, needs to be done early. I think there's no controversy about safety, but then other factors, for example, the recovery trial, which was given up, there 92% of those patients were also being administered steroids. And so there seems to be some bad interaction between application of steroids with convalescent plasma. And the more that we've sort of delved into the data, the more complicated the situation has become. And yet there are other RCTs, where there's extremely strong efficacy signal. And so I would say that it's something that, after some trials and tribulations, the FDA landed in the right spot with this, which is the EUA is fairly narrowly specified for early application of relatively high titer convalescent plasma. And depending on whether we end up being in real trouble with variants, then kind of locally sourced, where local means not only geographically, but also time-wise, source convalescent plasma could rise in importance. But, it's been an incredibly complex matter. One last thing to say is Microsoft, what the heck is Microsoft doing with this? And what happened there was, we had successfully worked with the CDC on the self-triage, self-assessment protocols and bots, and the whole tech industry helped distribute that. And so there was a request to work with FDA on doing something similar to encourage people to find out if they would be viable donors of convalescent plasma. And when we got involved in that, it seemed like a straightforward application of our technology. And we quickly found out that, the plasma, the fractionators in the pharmaceutical industry and the blood banks and the American Red Cross and the White House Coronavirus Task Force, they don't all exactly get along. And Microsoft found itself by accident as sort of the neutral intermediary. And I think our last phone call, Eric, I was frankly trying to get advice from you how to extract myself from the situation. And of course, we couldn't drop the ball on that. And here we are about 570,000 patients later.
Yeah, it's amazing. Yeah, when just the recovery was published as a preprint yesterday, and really, unfortunately, not showing the benefit that many of us had hoped to see. Let's go over to Christian, and maybe can you start the discussion on the variants? That's obviously a principal concern of ours right now. I know it's going to be a topic of 60 Minutes this Sunday, and maybe you can talk us through about what you're thinking about the major variants and the overall topic.
Sure. I think the first thing is probably to just identify what do we actually mean when we talk about variants, because I think there's a lot of confusion about variants. Which ones should we care about? Which ones shouldn't we care about? And the fact is that SARS-CoV-2, like all other viruses, mutate all the time, which means there are variants, if you want to call them that arise all the time. And the vast majority of these we don't care about. The trick is to figure out, of all the ones we don't care about, which ones should we actually care about? And I think as of late, there's been a lot of confusion around that, because there are three main ones. There's the one first identified in the UK, B.1.1.7, one first identified in South Africa, the B.1.351, and then the one first identified in Brazil, which is P1. And what we have observed about all of these is that given a set of selection pressures and some constellation of mutations, there are versions of SARS-CoV-2 which might have a higher fitness, might essentially spread better in a population than other versions of SARS-CoV-2. And that's exactly what we observed with the variants. And we have on the one side, again, the B.1.1.7 from UK, for example, is inherently more transmissible. and was simply selected for the purpose saying, well, the virus that transmits better in a population is going to rise in frequency. And that's exactly what we have seen from that. And we are now seeing that being widespread all across the world, including here in the United States. It is becoming widespread. The other two, I think, are a little different. I think they originated, emerged in areas of the world where you have very high attack rates, so they're called. They're very big outbreaks in the spring of last year. And these variants have now found themselves very successful in these environments. And I think the selection pressure there, while it might also be transmission, I think might have to do more with their ability to cause, for example, slightly higher rates of reinfections, because they have some immune evasion properties. It's never complete. It doesn't mean that if you have been infected with vanilla SARS-CoV-2, everybody will now be affected by these or infected with these new variants. But I do think that it's a different mechanism in which we have seen these actually become dominant in the regions where they're first identified.
So I guess to say the B117, you and a whole bunch of collaborators just put out a paper, and I know it's preprint, I know it's in press, is going to become dominant in the United States. And will it outrun these other variants that are associated with the immune evasion, like the P1 and B1351? And also, are you confident that we'll get through this?
We will get through this, but let me make that clear. But getting through this means we actually need to do something actively, right? I mean, Texas is done with COVID-19, but I can guarantee you that COVID-19 is not done with Texas, right? That's not the approach you want to follow here. I think B117, I do think, will in the short term become the dominant one in the United States over the next month or two, maybe. How it's going to compete with these other variants, we don't really know. And it also really depends on immune status in the population. But if we look at the other ones in the United Kingdom, they don't seem to be competing very well with B117. B117 has won out there. So I think we're going to see the same occurring here. And what I think is really important, and this is what our paper shows, is that we would like to think of the United States as being special, and all the stuff that happens in other countries are not going to happen here, right? But here's the sad truth, is that we aren't special. If we look at the growth properties of this variant in the United States, it's exactly the same as we see in the United Kingdom, in Portugal, in Denmark, in Jordan, and many other places. So yes, we should expect this to become dominant pretty soon, and we'll see in rising cases as a result of it.
I wonder, Akiko, if you could comment about the immune escape of the variants and the potential of a variant that we're starting to get a sense of in New York, which combines the features of B117 in the UK and adds this E484 onto it to make it kind of the worst case scenario potentially. What's your sense about how the vaccines will do with these variants that have some immune evasion property?
Yes. The variants that you're talking about clearly has some evidence that are reducing the ability of immune responses generated by the wild type virus or the vaccine to confer protection. So that is really a concern. And then, by combining, so these mutations that are arising that seems to escape the antibodies arising in multiple places, independently. And that again shows that, these things are being selected because they're able to infect people, even if they had some level of existing immunity. And so I do worry about these variants arising, both with respect to increased transmission rate as well as potentially, making these vaccines less effective. There is still cross-reactive immunity generated by most of these vaccines against the variants, and most of these vaccines appear to do a really good job of preventing severe disease and lethal diseases. So it's still very important to take the vaccines, no matter, what variants are out there. And hopefully vaccines that incorporate the variant sequence will also become available to really provide that, really high effective protection against these variants.
And Angie, you also comment about this topic and also that, not much, Peter brought up the T-cell tests that they've developed with adaptive. We don't really think about that when we think about immune escape. It's all kind of the neutralizing antibody story. Can you pull that together?
Yeah, so this is something I think about a lot actually, because I think that people have assumed that neutralizing antibody function is a correlate of protection for this virus, and it's really not. We don't actually have good correlates of protection yet. So clearly, T cells are playing a role. One thing that frustrates me about the entire variant conversation, especially with regard to immune protection, is the constant focus on mutations in the receptor binding domain of spike. That's not the only thing that determines if the immune system is going to effectively target a viral infection. That's also not the only part of a virus that determines how sick you're going to be. There are 25 other proteins encoded by SARS coronavirus 2. There are mutations throughout the genome in many of these variants that are different from one another. We spent so much time focusing on these mutations that abrogate neutralizing antibody binding that we haven't paid attention to any of these other mutations and any of these other genes that can also have an impact on pathogenicity and virulence. So I think, as well as immune protection from things like T cell epitopes, for example. So I think that, we need to have a little more humility in our public discussions about this significant. Certainly completely agree with Akiko and with Christian that we are seeing evidence of convergent evolution. We are seeing that some of these variants, very clearly the mutations, are providing some kind of fitness advantage or immune evasion advantage, but there's still a lot more that we need to know about these other mutations that seem to be arising.
Yeah, to your point, the receptor binding domain has been kind of eccentric thinking, and just today, there's been a couple of reports about the importance of the N-terminus domain and antibodies to that. Well, there's a question here from Gerald Powell at Salk, and he says to Peter, is anyone using BERT to define healthy people and find multigenic disease susceptibility? Is this respect with the advantage of having 2 terabytes memory on the TPU as opposed to the 32 gigabytes of the V100 or 512 gigabytes with the N V-link? So I'll turn that to you, Pete.
So first off, I guess the second part of the question is really touching on different computer silicon architecture choices, and those are important. But I don't think they make a meaningful difference, broadly speaking, at least at the scales. So the number of companies or entities outside of China that are experimenting with these hyperscale models is exactly 2. It's Microsoft and Google, and these involve in both companies multi-billion-dollar silicon and data center investments in order to build the size of the computing machines in order to experiment with these things. And in both cases, when you're investing that much, you have to have some potential return on investment that you're looking at. Not that it isn't speculative, but there has to be some story there, and so that means that the actual amount of progress that we've made and what we are seeing isn't fully disclosed. Having said that, there is a desire to open these things up more broadly, and even, for example, in the work with Adaptive, the ImmuneCode dataset, we're trying to publish all of the raw data openly in order to facilitate research. But especially with the language models, what we're finding is that we can't quite control what they say. And they can say things that would be harmful or embarrassing. And so that right now is something that, until we have a way to control that, would have to be handled very, very carefully. On the first part of the question, so we think that the potential to, for example, to use extremely large BERT-style models to drastically accelerate the molecular dynamics stage of, say, drug discovery is very significant. And we're talking maybe, up to 5 or 6 orders of magnitude acceleration. And so that's something that not just these two companies outside of China, but several companies within China, I think, are pursuing pretty aggressively. And I don't know that anyone can say that they will know where that will end up, but in a way, those problems are going along with all the other kind of commercial potential of these neural models.
Great. Thanks, Peter. This is to Angie from Michael's scheme and others on the panel. Moving away from the spike protein, what do you think about mutations of ORF8 and a possible connection to increased replication leading to higher viral load in the new variants?
So I think the ORF8 thing comes from a couple different things. So there's a stop codon mutation in the B117 that induces a premature stop codon in ORF8, and that results essentially in probably a non-functional ORF8 protein being expressed. Previously, during the SARS classic epidemic, mutations in ORF8 were associated with lower virulence, and that was a theory of how that was able to spread more effectively because it wasn't making people as sick. Now, that's just a theory about how B117 might be spreading. I don't think that alone is the secret to increased transmissibility. It's not really consistent either with the reports that B117 is more virulent, although, I'm not entirely sure what I think about that, considering that that's just an increased risk of hospitalization and death. It's not necessarily a mechanism by which the B117 variant is actually more pathogenic. So, the jury's out. I think that certainly there could be an effect one way or the other from a premature stop codon in ORF8, but we just haven't really done those studies. That would need to be done in animal models, and that type of mechanistic work takes time.
All right. Christian, do you think the B117 has increased lethality, which has been noted but not certain?
I do. I would say the jury is still out. I totally agree on that. But given everything we see with potential longer infectious periods, potentially higher viral loads, we're clearly seeing increased transmissibility. I think the most parsimonious explanation for the observations that it leads to higher rates of hospitalization and death, which we have seen not just in the UK, but in Denmark and other places too, I do think it's a real effect. What does that come down to exactly in terms of the mechanism of the virus? I don't know. I think probably some of the spike mutations play a role, but I'm pretty convinced that other genes play a role here too. Like we talked about interferon previously, for example, NSP6 and other mutations we find there, I do think could play an important role in determining that as well. I don't think it's just a spike, if it's the spike at all.
Right. Well, I have to say, I could go on with this panel. This is very sad for me to end this panel because this is the dream team that we assembled. My favorite immunologist, virologist, AI scientist, and Christian all in one time. You just can't beat this. So thanks so much for all of you for joining today at the Spectrum. It's really been intellectually stimulating, and I'm sure we'll come up with further synapses to explore ideas together. And thanks to all the participants for joining and for asking good questions. And just in closing, this is the end of 2 days of a lot of life science, a lot of drug discovery, a lot of cutting edge stuff, especially here on the pandemic in this last panel. We wanted to thank our sponsors. Diamond sponsor with Illumina, which was incredibly generous. They have been for years in our future of individualized medicine. Platinum sponsors Alexandria Real Estate Equities, AbbVie, Ambrx, and Dexcom, and our gold sponsor, BioMarin Pharmaceutical.